right, so welcome back. This is going to be our first lecture for our unit on DNA. So today we're going to talk a little bit about the history as well as the structure of DNA. So just a quick review about some of the things that we've talked about so far. Chromosomes are made up of DNA and protein. So chromosomes, which are located in our bodies and our cell's nucleus, is made up of DNA and proteins. On those chromosomes, so on our chromosomes are small segments called genes. And genes are segments of the DNA that code for a specific trait in our bodies. So in our body, we have chromosomes made up of DNA and proteins, and segments of those chromosomes are called genes, which code for a specific trait in us. Now, when we're talking about DNA, we have to go back a little bit in time. So it's actually not all that long ago, 1950s. So we're thinking prior to the 1950s, scientists did not know whether the genetic material inside of humans and living things was DNA or protein. They knew that we had something in our body that was responsible for passing on our traits and our genes, but they didn't know if that molecule that was responsible for passing on our traits was DNA or if it was protein. So there were three main groups of experiments that were done to help prove that the genetic material was DNA. So we're going to start by going through those three main experiments. So the names of the individuals that did these three experiments, the first was Frederick Griffith. The second group of experiments was done by a group of scientists, Avery, McCarty, and McLeod. And the third group of experiments was done by a duo, and their names were Hershey and Chase. So we're going to go through just a summary of these three experiments. So first we're going to go through Griffith's experiment. Fun fact, Griffith was not actually looking to try to find what the genetic material was. He was looking for something else, and he kind of stumbled onto his findings and questions about DNA. He was working with the bacteria Streptococcus pneumonia, which was a bacteria that caused disease in soldiers. He was looking for a cure to help cure disease in soldiers during the war. That was his initial experiment, was trying to help cure this disease in soldiers. So he was working with Streptococcus pneumonia, which caused pneumonia in soldiers. He was trying to cure them. And he knew that there were two different strains of this bacteria. There was the S bacteria, and the S bacteria was pathogenic, meaning it makes you sick. And the R bacteria was non-pathogenic, which means you did not get sick or did not cause sickness. So he was working with two different types of bacteria. And in his experiment, he had several setups. And his setup A, he went ahead and injected the mice with pathogenic bacteria. And this is what we call a control. He kind of had an idea. He knew it was going to happen. If you inject a mouse with bacteria that makes you sick, of course the mouse is going to get sick, and it died. And that's what he expected. He expected the mice to get sick, and he expected it to die. And that's exactly what happened. In his experiment B, this was another control. He injected the mice with non-pathogenic bacteria, so the bacteria that did not cause sickness. And of course, if you inject a bacteria that does not cause sickness into a healthy mouse, the mouse should not get sick because the bacteria does not cause sickness. And then he started doing his two tests. In his third experiment, he used heat to boil the bacteria that cause sickness. So he killed the bacteria that cause sickness. And if you kill the bacteria that causes sickness, it can't do its job anymore. He injected it into the mice, and the mice lived because he killed the bacteria. If they're killed, they can't make you sick anymore. In experiment D, something weird happened, and this is the one we're going to mainly write down. He did this weird experiment where he took heat-killed bacteria that make you sick. So he took killed S bacteria that typically do not cause sickness. So typically, they do not cause sickness. He proved that in experiment C. He mixed that killed S bacteria with live R bacteria, or bacteria that don't make you sick. And normally, this type of bacteria does not lead to sickness. He proved that in experiment B. He noticed when he mixed them together, so he mixed them together, he injected it into the mice, and it equaled death. And he was curious about that. How did a dead bacteria that would make you sick, but usually doesn't, and a live bacteria that does not make you sick, how, when you mix them together, how exactly does that mice get sick and die if the two things alone don't cause death? And the only thing that he could sum it up to was this theory called transformation. And that's the idea that there was something inside of this killed S bacteria, something that causes sickness, that was able to travel into the live bacteria and cause it to turn bad. So his idea was that something inside of this bacteria moved into the live bacteria that doesn't make you sick. Sorry, that was my dog. And made them deadly. And he called that thing the transformation factor, which would be the factor inside of the S bacteria that causes it to make you sick. So that was the summary of his experiment. He didn't know what it was that moved. He just knew there was something inside of that killed S bacteria that moved into the living R bacteria that made it go deadly. And he called that thing that moved the transforming factor. So that's Griffith's experiment. He, as well as some other scientists, knew that something inside of it had to transfer the genetic material. There was some type of gene that caused sickness in that S bacteria that was able to move into the R bacteria to make it deadly. But again, he didn't know if it was DNA or if it was protein, and that's where our next group of scientists picked up. So Avery, McCarty, and McLeod, they took on this question of what is the transforming factor? What is the transformation factor that moved from the S bacteria to the R bacteria? Or, in other words, what is the genetic material? This is what they were looking for. So they did two experiments. And what they did is they worked with the same bacteria, the Streptococcus pneumonia bacteria, and they knew the results from Griffith's experiment, and they did two experiments of their own. So the first one that they did is they took protein from the bacteria, they isolated the proteins from the bacteria, they injected it into a mouse, and when they did that, nothing happened. Their idea was if they were to isolate the two factors and inject them into mice, whatever one had the gene would cause change. That was their idea. So they took all the proteins out, purified it, injected it into the mice, and nothing happened. So they did the same thing with the DNA. They isolated the DNA. They took all the DNA out and they injected it into a mouse, and what they noticed is transformation happened. Something changed. And that led them to think, well, it's probably DNA that is the genetic material. But they wanted to be double sure. So they did experiment 2. And in experiment 2, what they did is they destroyed proteins. So they took away proteins. When they took away all of the proteins, they still saw transformation. And what that means is that the genes were still there. So when they took away the proteins, there were still genes. There's still that transformation factor. But when they took away the DNA, there was nothing. Meaning, when they took away the DNA, they took away the genes. So that was Avery, McCarty, and McLeod. Now, in science, if one group of scientists does an experiment, everybody's not going to just believe the results. It has to be done multiple times. So that's why Hershey and Chase did another experiment to try to prove that DNA is the transformation factor, that DNA is in fact the genetic material. So Hershey and Chase, they did what is called the blender experiment, and they worked with what is called bacteriophages. Bacteriophages are viruses that infect a bacteria. So bacteriophages are viruses that infect bacteria. And what they did is they had their virus here. And what they did, viruses are made up of only 2 components. They are made up of protein and they are made up of DNA. So what they did is they tracked the DNA and the protein in how this virus worked. So the virus is up top and it infects a bacteria. So what they did is they used radioactivity. They had 2 setups. So they knew that the genes were passed from the virus to the bacteria. So what they did, I'm going to change colors here to red. In experiment 1, they labeled the protein with radioactivity. So they labeled the protein with radioactivity. The red represents radioactivity. In experiment 2, they labeled the DNA with radioactivity. And then they let the virus do its job. They let it infect the bacteria. And their idea was, if protein is the genetic material, then it will go into the bacteria. If DNA is the genetic material, then it will go into the bacteria in experiment 2. And what they noticed is that in experiment 1, nothing happened. The virus did not make the bacteria radioactive, meaning the protein was not transferred. However, in experiment 2, the virus made the bacteria radioactive, meaning DNA was transferred. Therefore, DNA is the genetic material. So Hershey and Chase came to the conclusion that DNA is the genetic material by tracking what was transferred from the virus to the bacteria by labeling it with radioactivity. Okay, so this is how we figured out DNA is the genetic material. Now we're going to go into a little bit about the structure of DNA. So again, this goes back to scientists doing their work in the lab. Two individuals, Wilkins and Rosalind Franklin, worked to take X-rays of DNA. So Wilkins and Franklin, they worked in their lab taking X-rays of DNA. So they would take tons and tons of X-rays of DNA. And they got this image here. This is an X-ray of DNA. So I'm going to go ahead and draw this over here because you'll want to be able to recognize this image. This is an X-ray of DNA. What this X-ray was able to tell them is the structure of DNA. They were able to look at this image. Specifically for them, it was photo 51. They looked at it, they worked backwards, and they were able to figure out that DNA is a helix. The key memory for these scientists, Franklin took X-rays of DNA, and that allowed them to figure out DNA as a helix. So DNA is the molecule of heredity, meaning it contains genes, and those genes are passed on to your kids. And DNA stands for deoxyribonucleic acid. And DNA is double-stranded. It's a nucleic acid that determines an organism's traits by controlling the production of protein. So DNA, deoxyribonucleic acid, is a double helix. Its job is to contain genes that are going to make proteins that give us traits. So inside of DNA, DNA is made up of millions of nucleotides. You can remember this from way back when we did biochemistry. I feel like I spelled that wrong. There we go. And nucleotides contain 3 parts. They have a sugar. It's a 5-carbon sugar. They have a phosphate group and a nitrogen base. So if we were to draw out a nucleotide, I like to draw the sugar first. So this is my sugar. 5-carbon sugar should have 5 points. That is connected to a phosphate. Phosphates are a little circle with a P in it. And then there is a base. And the base is a nitrogen base because it contains the element nitrogen. So we have 2 more scientists. I believe these are our, maybe there's one more, almost our last 2. And their names are Watson and Crick. Now, Watson and Crick are credited with discovering that DNA is made up of nucleotides that are held together by nitrogen bases. So they found out that DNA is a double helix. And it looks kind of like a spiral staircase. Now, I'll tell you, these 2 guys, they're kind of tricky. They sat in on a secret meeting with Rosalind Franklin. And that's where they saw the images of the X-rays. And they kind of stole that and published the double helix information before Franklin could. So Watson and Crick get credit for discovering the double helix, but they didn't put in nearly as much work. So they stole her work, they got a Nobel Prize, and she died because of exposure to so much radiation for taking the x-rays. She died for her work because she was exposed to so much radiation, and they got the Nobel Prize for discovering the double helix. Kind of a weird situation in science. So again, nucleotides have 3 parts. The sugar is deoxyribose. Now, if you notice, if we were to go back up, DNA stands for deoxyribonucleic acid, and the sugar in DNA is deoxyribose, so DNA's name comes from the sugar. There is a phosphate group. And then our last is a nitrogen base. The nitrogen bases, even though I often draw them as rectangles, they are actually ring structures that could have 1 or 2 rings. So each backbone or side, so when I'm talking about, these are going to be the backbone or the sides of the DNA. They're made up of alternating phosphate groups and deoxyribose sugars. Therefore, it is referred to as the sugar phosphate backbone, because it's made up of sugar and phosphate. Now, in the middle, the rungs of the ladder are going to be made up of those bases, those nitrogen bases. And the nitrogen bases are going to be bonded to each other in the middle of the ladder. So this activity here, it says connect the backbone of the DNA molecule with covalent bonds and the nitrogen bases with hydrogen bonds. So if this is my DNA molecule, this sugar is going to be attached to that phosphate. The same over here, this sugar attached to this phosphate. Those are covalently bonded together, so it's a solid line. In the middle, the DNA is attached with hydrogen bonds, which is a dotted line, because hydrogen bonds are very weak. And we'll talk about in our next video why it's important that bases are weakly bonded together. Now, there are 4 different types of nitrogen bases. There are 4 different ones. There is adenine, which we're going to represent with the letter A. There's thymine, which we're going to represent with the letter T. There's cytosine, which we're going to represent with the letter C. And there is guanine, which we're going to represent with the letter G. So there are 4 bases, 4 different nitrogen bases, and there are 2 different types or classes of them. Pyrimidines are single ring sugars or bases. So they are single ring bases, meaning they only have 1 ring. And the way that you can remember this is long word, short structure. And the way that you can remember which bases are pyrimidines is because they have a Y. Pyrimidines have a Y. So PY, THY, CY. So thymine and cytosine are pyrimidines because they have a Y. Purines are double-ringed bases. So purines are double-ring bases. The way that I remember that is short word, big structure. And again, they're the ones that do not have a Y. Now, in DNA, bases always pair together predictably. Adenine always pairs with thymine. So A and T always join together, and C and G always go together. And these are called complementary base pairs. So these are called complementary base pairs. The way that I remember this is that C and G are both curved, so they go together. So C and G letters are both curved, so they go together. That's how I remember that one. And here's our last scientist. His name is Chargaff. Chargaff stated, based on the base pairing rule, the amount of A is always equal to the amount of T, and the amount of G is always equal to the amount of C. So, summary, what he said was A equals T and C equals G. And you know that because they always pair together. If there are 5 A's and A is always paired to T, there has to be 5 T's. So A equals T, G equals C. A little mnemonic device to help you remember A, T, C, G. You can remember the curved thing, the C's and G's are curved, or you can remember apple trees, A and T, and collard greens. Totally up to you. Whichever one floats your boat that you can remember better. So apple trees, AT, collard greens, GC. And this is going to bring us to a simple math equation that you may see. So number 1, if one strand of DNA contained the sequence ATCGGAT, what would the other strand be? So this is essentially asking you to figure out what pairs with what. So we know that A always goes with T, T always goes with A, C goes with G, G with C, G with C, A with T, and T with A. So you just find the opposite. Number 2, if a segment of DNA has 100 total nucleotides, 30 are A, figure out the others. So the way that I like to do this only has the 3 listed. I'm going to go ahead and write out my A as well. So I always write out my 4 bases, A, T, C, and G. And then I fill in what I know. I know that 30 of them are A. And we said earlier that A is equal to T. So if A equals T, that means that T also has to be 30. When I add those up together, I get 60. And they said I have 100 total. That means I have 40 left. And I know that G and C have to be equal, so I have to divide that by 2. And that gives me 20 for C and G. So again, the way that I did that, I have 30 A's. A is equal to T, so there has to also be 30 T's. Together, that only gives me 60 bases. And I started with 100, so I do 100 minus 60, and that leaves me with 40 left. I have 2 bases left, C and G. I know they're equal, so I divide by 2, and I get 20 each. All right, so there's going to be some practice on this before we do our next set of notes.